Hello everyone, welcome back to another session of regeneration biology. And today's topic we will learn about the future implications of regeneration in mammals. So in today's class, our focus would be to understand what are the future angles or the future of regeneration research using mammals, or what are the implications that going to affect or going to bring about a step closer to human tissue regeneration, because humans are also mammals and we use mice and other organisms as a model to adopt the strategy of regeneration in humans. So that is the overall theme of the so-called uh, regeneration biology or regenerative medicine, etc. So fortunately or unfortunately, the mammalian model that is extensively used in laboratories are mice. Of course, rat, guinea pigs, rabbit, or even monkeys are also there, but majority of the research is oriented around mus musculus, that is uh, normally called as laboratory mice, and that is what we use it for our regeneration studies also. However, this animal do not have regenerative ability, so that is why it becomes little challenging to study a proper regeneration or a actual mechanistic study of regeneration becomes little difficult in mus musculus model. So we will see what are the future implications that hold, and let us quickly see an overview. The future research into mammalian regeneration holds the promise of transformative medical advances, potentially enabling repair or replacement of the damaged tissues and organs, particularly those with limited or no regenerative capacity, such as heart or brain. We do have regenerative capacity of hair, nails, skin, etc. And also one major organ we regenerate is liver. So we know that very well. However, other complex organs such as brain, spinal cord, retina, etc. do not have, or even heart, do not have proper regenerative capacity. That is becoming one of the major target for regeneration biologists. So understanding the molecular and cellular mechanism underlying regeneration in other animals and identifying factors that constrain regeneration in mammals can lead to development of novel therapeutic strategies. Having said that, very well regenerating models such as zebrafish, axolotl, etc. are effectively used to copy this mechanism or simulate this mechanism in mammals and have got promising results. What it tells us is that the regeneration mechanism or the way in which regeneration can take place do not change too much across different species. All we see is in some species such as fishes and frogs have these mechanism in place, properly orchestrated manner they are in place, whereas in mammals, say mice or humans, despite having all the options of proper regeneration, despite having all the genes responsible for regeneration, their expression is not adequate or their temporal regulation is not efficient. This has led to poor or lack of regenerative ability in these mammalian models. So let us see how can we revolutionize the regenerative medicine. So treating the currently untreatable conditions often look forward to regeneration biology. So the ability to induce regeneration in mammals would revolutionize the treatment of conditions like heart failure and neurological injuries such as stroke or spinal cord injury, etc., where the current therapies are very limited. So overcoming the organ shortage, that is another approach which we have to go for this regeneration biology, because we do not have adequate organ, whether it is a cultured organ or we have the donor organ or a cadaver organs. If heart organ regeneration could be achieved, it could alleviate the strain on the organ donor pool. So developing novel therapies, that is another approach of the immediate prospect of regeneration biology, is the researchers can develop targeted therapies to stimulate tissue repair and regeneration by understanding the molecular pathways involved in regeneration. That is another approach. Now, looking into the unlocking the secrets of regeneration, we will find a few interesting facts. That is understanding the blastema. We know what is blastema by now. I will not go into the details, but all we know is blastema is a prelude for a normal regenerative response. If there is no blastema, you end up getting a wound healing. You do not end up getting a regeneration. The blastema is specialized tissue that forms during regeneration in some animals, is a key of research of in the regeneration biology field. Studying mammalian blastema, like those found in regenerating rodent digits or ear pinnae, two models are used for regeneration in mice and rodents in general, is either you cut the toe tip or you cut their ear pinna. Both regeneration can be studied. They can provide insights into how these structures are formed and how they drive regeneration or how they do not drive regeneration properly. So, identifying therapeutic targets by analyzing the molecular signals and cellular events during regeneration, researchers can identify specific molecules and pathways that could be targeted for therapeutic intervention. So, the understanding factors also can be a limiting point of regeneration. That is, the mechanisms that limit regeneration in mammals, such as wound contraction. We have seen it. The wound, soon after wound is formed, they contract because of the cytoskeletal and other mechanistic force that comes, or mechanical force that comes into picture. Wound contraction, and then comes the immune responses and changes in the microenvironment. They are also active research. Can we prevent the wound contraction? Can we prevent unwanted immune cell migration, etc., or excessive deposit of collagen, etc.? If you can we think of overcoming? So these are all some active areas of research. Modifying these factors could potentially enhance the regenerative capabilities in non-regenerative animals such as mammals. So, pushing the boundaries of epimorphic regeneration. We know epimorphic regeneration is the most desirable regenerative mechanism a species can have. So, can we push the boundaries of the epimorphic regeneration? Extending the regeneration beyond the limbs. While some mammalian tissues such as skin, hair exhibit their regenerative capabilities, the focus is on expanding regeneration to more complex tissues and organs. So this is an active area of research. This could involve stimulating the formation of blastema somehow. Naturally doesn't form, can we stimulate in other tissues, and using techniques like tissue engineering to promote the regeneration. So can we think of engineering a regenerative tissues? So researchers are now exploring using biomaterials and stem cells to create the scaffold or 3D printed organs that can be implanted to promote regeneration. So these are also an active area that is going on in regeneration biology field. And harnessing the body's natural healing process is the most attractive prospect, simply because we have discussed many times, even though you can make an organ in the petri dish, but if its blood vessels or its proper angiogenesis do not intercalate with the recipients or the host's blood vasculature system, the future of this organ is in grim. So even though it is a perfect organ, the body did not nurture it, then it is less likely going to be a successful organ. Hence, it becomes very important harnessing the body's natural healing process. If you can allow the healing or the fixing to happen side by side in the same organism itself, although it may be time consuming, although it may be cumbersome, but you don't need to worry about it, that there will be enough oxygen supply or blood supply. That is why the natural healing process becomes more attractive. Researchers can develop synergistic therapies with these processes to enhance tissue repair and regeneration by understanding how body naturally heals and regenerates. So that is the ultimate goal of tissue regeneration biology, is about natural healing. All we need is some triggering or a stimulation in those animals which do not regenerate effectively. So your goal is to get into that era. So now, as any other topic has, we have discussed many times that the ethical and societal implications are also very important. It is so important that sometimes even the research itself can come to a U-turn, or it can come to a still state, simply because of you are not meeting the ethical considerations of that particular research. Addressing the safety concerns. As regenerative therapy becomes more advanced, addressing potential safety concerns such as the risk of tumor formation or other unintended consequences, it is crucial. Any approach you adopt, you have to be sure that this patient who received this treatment is not going to die of cancer now because of this treatment. It has happened in the past because of the gene therapy. I think it is done with to treat the hemophilia, the blood clotting disease. Those two treatment done on the children, they ended up developing leukemia. Hence, the gene therapy became a complete, standing still situation happened, because the approach can make them vulnerable to getting into cancer, because you are dealing with the cell proliferation. And whenever you see an unwanted cell proliferation in your body, that is a red flag for favoring, or it's a red flag for the organism, which means that it is favoring the formation of cancer. Ensuring the equitable access. This is another thing, that everyone should get access to such a technology. Regenerative therapy should be accessible to all individuals regardless of socio-economic status or geographic location. So it shouldn't happen such that the layman or a financially underprivileged people are deprived of. That is another serious ethical angle or societal implication angle that comes into picture. Considering the long-term impacts, the long-term effects of regenerative therapy need to be carefully considered, and the research should focus on developing therapies that have a lasting positive impact on patient health and well-being. In conclusion, the future of mammalian regeneration holds immense promise for transforming healthcare. Understanding the underlying mechanisms of regeneration and developing novel therapeutic strategies makes it possible to unlock the body's natural healing potential and revolutionize the treatment of a wide range of disease. That is why the future angle or the future implication should be met very carefully. Let us now look how effectively or how efficiently you can transform or modulate the so-called regeneration biology field using other mammalian models. Do we have other mammalian models that has some better or the best available regenerative capacity? So Van Bainum, a scientist, he explains that one model organism called spiny mouse is a fascinating model for studying regeneration. So by examining the unique characteristics of this animal, that is spiny mouse, which is normally seen in very dry and desert conditions, we will gain better understanding of the possibilities for developing therapies to improve wound healing in humans and increase the likelihood of full recovery. So this is the picture of spiny mouse, which resembles ordinary mouse, except that instead of hair, it has got spines on its back, and its tail also having some spines, and it has got excellent regenerative capacity. Instead of musculus, its its generic name is called Acomis, and it has got around 20 different genera. Many of them have regenerative capacity, not all of them. One of the commonly used one is Acomis kempi. So many species are there and normally seen in Africa, Brazil, etc. So let us see some few stories of regeneration in spiny mouse. A team of researchers at University of Kentucky and Cincinnati Children's Hospital is delving into deeper science behind how spiny mice can regenerate lost tissue using what they learn to trigger regeneration in other types of mice. One other types of mice fails to do how these spiny mouse is able to do this. So advances which one may have. So as you keep looking into the research, which one day we will be able to translate the information you learn from spiny mouse be put into for humans use. Adult laboratory mice heal injuries with scar tissues, that is mus musculus, not spiny mouse, while the spiny mouse can regrow lost skin and regenerate musculoskeletal tissues including the spinal cord. So it can regenerate complex tissues. That is the beauty, not about just regenerating just some epithelium or skin or intestine, that we also have the ability. Making a complex tissue regenerate is more important. One of the pioneers scientist is Ashley Seifert and his research group have pioneered using spiny mice and other animal models to understand how complex tissue can regenerate, bridging the regenerative biology and medicine. So the knowledge from spiny mice is going to revolutionize the regeneration biology field. Let us see the science behind spiny mouse regeneration. A study from same group, Seifert's group, focused on cellular response to injury during tissue healing. So the study showed that how ERK signaling, ERK signaling, acts as a key switch in balancing the healing response, and they have demonstrated that the increasing and maintaining this type of signaling, increasing means more than usual and not just more than usual, sustained signaling, ERK signaling in the laboratory mice could trigger a regenerative response instead of scarring. So the message is, in spiny mouse there is ERK signaling active and persistent throughout the regenerative response, whereas in laboratory mice ERK signaling triggers and it disappears, and hence it is regenerating. It is not regenerating, but it is healing the wound with scar formation. Also, how specific immune cells respond to injury and direct tissue regeneration deserves a proper attention to understand the involvement or explore the involvement of specific immune cell types. The scientists looked at a specific type of immune cell known as macrophages. We have discussed many times that there are two types of macrophages, M1 and M2 macrophages. M1 are pro-inflammatory, M2 are anti-inflammatory. During fin regeneration class, we have discussed about that very effectively or very elaborately. So the macrophage which play a crucial role in regulating inflammatory response. Regulating inflammatory means it can make it pro and it can make anti depending upon which macrophage you are discussing. And inflammatory response by defending injured tissue against pathogens while simultaneously promoting tissue repair in both spiny mouse and lab mouse. Let us see further. The macrophage phenotype between these two models of healing, that is mus musculus and accumis, namely tissue regeneration or scar formation, they differ. Tissue regeneration in spiny mouse, scar tissue formation in mus musculus, that is laboratory mouse. While it is unclear the extent to which macrophages ultimately control different healing trajectories, the observed subtle and distinct macrophage signatures aligned specifically with the regeneration. It's just like people say, there is a saying that winners don't do different things, they do things differently. Like that, the macrophage is there in both, but the gene expression, the factors which are expressed in spiny mouse is different from that of laboratory mouse. Using identical injuries in both rodents, means mice, mus musculus and spiny mouse, the researchers studied signals released by macrophages communicating to nearby cells, telling them to rebuild all missing tissues. How does the building happens? During those processes, they also noted the cell's characteristics or the phenotypes, how the cells respond to the signals released by the macrophages. The study showed that the spiny mouse macrophages release distinct proteins partially responsible for reforming specialized tissues at the injury site and protecting the cells from the stress. That means it is helping the cell not to die, not to succumb to the stress, injury stress, and also make different types of cell because they release some unique factors. Overall, these experiments point towards the macrophages establishing a tissue microenvironment conducive for regeneration. Researchers examined and compared macrophages from bone marrow of both these species, that is, mus musculus and accumis. Then they used RNA sequencing to identify the proteins these cells secrete. RNA, we all know, it is ribonucleic acid responsible for the production of various protein. It's a molecule that carries genetic instruction from DNA to make protein. RNA is the intermediate. Researchers found that the macrophages from bone marrow responded differently on their own to a protein called interferon gamma, IFN gamma, which is a pro-inflammatory cytokine, and M1 macrophages can secrete that as well. A protein produced by cells in response to infection, and lipopolysaccharide, a molecule found in the bacteria, bacterial lipopolysaccharide, LPS, that stimulates the immune system. And because of the stimulation of the immune system, you will have plenty of interferon gamma that is produced at the site of, or they are present at the site of injury. In response to inflammatory cues, macrophages in spiny mouse release unique communicating proteins that promote the growth of new blood vessels, lymphatic vessels, anti-inflammatory activities, and tissue rebuilding that positively contribute to the regeneration. So, same molecule, it is there in mice also, but with interferon gamma, the spiny mouse does these steps that forms new blood vessels and other rebuilding activity of the tissue. To answer more scientific questions about spiny mouse, researchers have found that a specific type of protein that is called vascular endothelial growth factor C. Normal one is VEGF, we say, vascular endothelial growth factor. This is VEGF-C, is uniquely secreted by spiny mouse macrophages during regeneration. It also plays a multifunctional role encouraging the growth of new blood vessel and lymphatic vessels. Like I mentioned, the newly formed tissues future is only that much important or that much successful as the blood vessel or a lymphatic system is formed properly. Otherwise, the newly formed tissues future is in grim. Through specific antibody blocking of the same VEGF-C protein in the ears of spiny mouse, because that is used as a model. You cut the ear pinna and look at whether it is regenerating. Researchers noticed notable changes in the formation of blood vessels, lymph cells, and cell division. So they found that if you block it, you don't get a proper regeneration. So VEGF-C is needed. This led to the decrease in this blocking of VEGF-C led to decrease in the new hair follicle formation and increase in the inflammation, ultimately disrupting tissue regeneration. So means you block one factor, VEGF-C released by the macrophage, you can abort the regenerative ability in spiny mouse. Additionally, the researchers speculate that the enhancement of secretion of VEGF-C and other factors by macrophages during fibrotic wound healing may be contributing to a regenerative outcome in tissue healing. However, further experiments are required to confirm this idea. This is a smoking gun. You can prove this point, but one has to delve more deep to understand the exact mechanism. The study suggests that the macrophages in specific tissues throughout the body help direct and regulate the cellular repair program. So you may wonder how come one molecule can play a role. We should understand the VEGF-C is a master player provided there is its receptor and those cells respond in a different manner. Like I am giving a lecture now, maybe you are listening, maybe your pet animal also listening. You will understand what I am saying, but your pet animal will listen it as someone is speaking. It won't understand in as much clearly as a human being can understand. It simply means the ability to respond depends on which cell it is responding to that signal. Importantly, the results of this study show that the type of macrophage matters and suggest that the altering what one type of macrophage secretes could alter how tissues repairs itself. Of course, macrophages are important. What protein it is secreting is important, but it also matters that which tissues are responding to that secreted protein from this macrophage. Another spiny mouse regeneration scenario we can look. When comparing the tissue healing in laboratory mice and spiny mouse, it was observed in general that adult laboratory mice heal injuries with scar formation. That is, injury is common in both, but in another one species it is wound healing, normally reflected by scar formation. In another mice, spiny mouse, regeneration is scarless. Spiny mice are known for their unique ability to regrow skin, especially restore lost skin, restore functions of the severed spinal cord, and one of this Seifert's, the same scientist we are talking about, previous studies show that repair damaged heart tissue to maintain heart function effectively. It can regenerate even damaged heart tissue. The extracellular signal regulated kinase ERK, this we discussed earlier, is a major cellular response to injury during tissue healing. ERK mediated signaling acts as a key controlling healing process in spiny mouse. Researchers, the same group of researchers observed that ERK activity in response to tissue damage in both rodent species. However, ERK activity decreased in lab mice as initial wound healing phase and tissue inflammation resolved. So, like I told you, ERK signaling is there in every species and it is turned on in lab mice also, spiny mouse also, but the ERK signaling in mus musculus, normal mice, is turned off when the wound healing phase and the tissue inflammation phase is completed. In contrast, the ERK signaling was maintained in a diverse array of cells at high levels in spiny mouse as new tissue was generated. Remember, wound healing is fast and regeneration is slow. So, these mechanisms that is helping for regeneration should be there for a prolonged time. As part of the same study, the researchers applied ERK activators to the ear of the lab mice, like same ear pinna cutting, and normally doesn't regenerate in lab mice, but they put ERK signaling activators and generated the same regenerative healing they observed in spiny mouse. All they did was they prolonged the ERK signaling activity and they could get a better regenerative response. Researchers found that sustained high ERK activity is critical for regeneration. From this message we can learn, just like VEGF-C we saw, if you block it you don't get it. Same way, sustained ERK signaling can favor the regenerative response in lab mice ear pinna. That partially explains why some closely related species like spiny mice and lab mice may have very different cellular response to the same injury. It is quite surprising. In this spiny mice regeneration research, researchers asked whether poor regenerators, namely laboratory mice and even humans are not laboratory animals, but our interest in regeneration research is to help humans, have completely lost their ability to regenerate or still possess their latent regenerative traits. This is a $1 million question. If the latter hypothesis holds true, that means we still have got this ability to regenerate, it would have enormous implication for regenerative medicine, implying that a regenerative response can still be triggered in cells and tissues of poorly regenerating adult mammals and ultimately can benefit humans. So we should understand that this regenerative ability can be modulated across the species to get the ultimate or the most desirable regenerative capacity. So we will study about regeneration biology with a different topic in the next class. Thank you.